Good morning, everyone. Welcome to Lab Medicine and Pathology Grand Rounds. Today, I have the pleasure of introducing TBrown, who is a student in the medical scientist training program here at the university. And as part of their curriculum, they are supposed to present some of their work to a clinical department, and she has wisely chosen to present her work to us. Taylor has she is a graduate of Villanova University, where she majored in biochemistry and neuroscience, and she has been working in the laboratory of Rocio Gomez Pastor in the neuroscience department here. She's received several awards, actually, during her graduate work here. One, which is the Morris Smithberg Memorial Prize for Outstanding First-Year Student in Neuroscience. And then just recently has won the American Legion Brain Sciences Award, which is also given to really top-notch trainees in neuroscience. So she's already become quite accomplished. She hopes to defend her dissertation this spring and return to medical school in the summer. Today, she's going to talk to us about GFAP positive astrocytes in the ZQ175 mouse model of Huntington's and how that translates into human pathology. So, Taylor, go ahead.
Thank you. Thanks for that introduction, and thank you for the invitation to speak at this Grand Rounds today. I am going to turn my camera off, I think, just to preserve bandwidth, but I wanted you to see my face before I do that. Okay, so like Dr. Clark said, I am a sixth-year MD PhD student. I'm wrapping up my PhD soon, and then I am going back to med school in the early summer. And I'm planning to pursue a career in neuropathology. So I've been working with Dr. Chen for some neuropath experience, and I am excited to get to talk to you all today about my research in the Gomez Pastor lab. Okay, so my plan is to discuss Huntington's disease, or HD as I'll call it, and the role of astrocytes in HD. So I want to talk about my research as well, in which I'm looking at the spatial distribution of a specific subtype of astrocyte in HD and trying to determine what role they play in the disease. And then lastly, I wanted to talk a bit about the HD mouse models and the translatability of discoveries made in those mouse models. And I have no disclosures. Okay, so let's start with a little bit of background on HD. It's an adult onset neurodegenerative disease, and typically these patients are diagnosed in their 30s and 40s. It's a really devastating disease that's characterized by progressive motor, cognitive, and psychiatric deficits. Probably the most well-known symptom of HD is chorea, or irregular and unpredictable muscle movements. And this is characteristic in the early stages of disease, but later on, patients experience impairment of voluntary movements, coordination, and bradykinesia. The disease is fatal, with a median survival from motor onset of only 18 years. And there currently are no cures or disease-modifying therapies. Right now, patients can use tetrabenazine to reduce involuntary movements, but it comes with significant side effects, and it does nothing to stop or slow disease progression. HD is an autosomal dominant disease, and it's caused by a CAG trinucleotide repeat expansion in the Huntington gene. And this leads to a polyglutamine expansion in the Huntington protein, and the expansion causes mutant Huntington protein to be dysfunctional, unfolded, and aggregated, and we call it MHTT for mutant Huntington. The normal function of the Huntington protein is not fully understood, but some broad biological functions have been uncovered. And Huntington is expressed throughout the body, but at varying levels depending on cell type. Mutant Huntington preferentially affects medium spiny neurons, or MSNs, of the striatum. The striatum is a brain region that controls movements and some forms of cognition. And you see HTT aggregates all throughout the striatum in HT mice and in patients. So here is an image of HTT aggregates in medium spiny neurons. It's still unclear exactly why MSNs are so vulnerable, but you can see here the clear and selective degeneration of the striatum in this image of an HD brain. Most of the HD research so far focuses on MSNs, but today I'm going to be talking about astrocytes, which are a type of glial or non-neuronal cell in the brain. Degeneration of MSNs in HD is accompanied by an increase in neuroinflammation and astrocyte pathology in the striatum. Since astrocytes have so many roles in the brain, which I will get into in a few slides, it's been hypothesized by others in the field that astrocyte dysfunction may cause or contribute to MSN degeneration. And this idea has been partially tested by doing cell-specific mutants. So a variety of studies have attempted to either express or knock out mutant Huntington in only specific cell types. And this is a chart summarizing many of those studies and showing which cell types were manipulated. It's a lot, so don't worry about the whole thing. I will summarize the key relevant findings. First, mHTT exclusively expressed in MSNs only partially recapitulates the HD phenotype. And notably, it does not produce motor deficits. However, pan-neuronal expression of mHTT does produce motor deficits. So it's hypothesized that an interaction between cortical and striatal neurons are important for disease pathogenesis. Expressing mHTT only in astrocytes also only recapitulates part of the HD phenotype. Those mice showed lower body weight and some motor deficits. But it also produced the transcriptional changes seen in HD astrocytes that cause astrocytic dysfunction, indicating that that part of HD could be cell autonomous. Now, on the other hand, reduction of mHTT only in astrocytes or only in MSNs did not rescue the HD phenotype. So clearly, the HD phenotype has components that are cell autonomous and non-autonomous for both astrocytes and neurons. And all of this is evidence that HD is caused by complex neuron glia dysregulation and that astrocyte pathology plays a role in the disease. However, the extent of that participation has not been fully uncovered. And part of the reason why it's been hard to uncover is because astrocytes play a role in lots of different processes and they're heterogeneous. Okay, so astrocytes are a really cool cell type. They do a lot. They are part of the tripartite synapse, so they regulate synaptic activity. They're also part of the blood-brain barrier. They're involved in synapse formation and phagocytosis, as well as energetic support. They clear glutamate and potassium. They clear glutamate from synapses and they maintain potassium and glutamate homeostasis. They also interact and communicate with other glial cells, and they respond to injury and inflammation. Basically, they're keeping neurons alive. And I really want to emphasize that astrocytes are involved in so many functions, and many of those functions play a role in HD. Astrocytes can release gliotransmitters, which are like neurotransmitters, except released from astrocytes, and they can have direct impacts on synapses and influence circuit outputs. Intracellular calcium elevations can cause the release of gliotransmitters, so a lot of astrocyte researchers will look at astrocyte calcium signaling as a measure of astrocyte function. And so that's been done in HD, and astrocyte dysfunction was seen. To measure calcium signaling, you use a fluorescent calcium indicator, and then delta F over F is just the change in fluorescence from baseline. So you can see here that there is significantly reduced change in fluorescence in the HD mice compared to the wild types, meaning that striatal astrocytes in HD have reduced spontaneous calcium signaling. And this is thought to be due to reduced internal calcium stores. HD astrocytes also have reduced Cur4.1 and GLT1, which leads to increased extracellular potassium and glutamate. And this contributes to hyperexcitability and possibly excitotoxicity of MSNs. The previous slide showed data from a fast progressing mouse model of HD, and we in the Gomez-Pastor lab use a more slow progressing mouse model, and I will go into both of those later, but we have shown the disrupted calcium signaling in our own lab. We use the calcium indicator GCAMP6F, and you can see an outlined astrocyte here, and watch as it changes in fluorescence over these 4 seconds. And here you can see that HD astrocytes have similar, smaller calcium events compared to wild types. Okay, moving away from astrocyte dysfunction. Like I said earlier, astrocytes can also respond to injury or inflammation in a process called reactive astrogliosis. During astrogliosis, astrocytes upregulate intermediate filaments in the cytoplasm and they undergo hypertrophy. And this is a process that involves both biochemical and structural changes to astrocytes. Traditionally speaking, glial fibrillary acidic protein, or GFAP, is thought of as a reactive astrocyte marker. GFAP marks intermediate filaments in astrocytes, and it is particularly enhanced in response to injury or inflammation. But GFAP's exact function remains poorly understood. It's possible that it might be involved in astrocyte dynamics as it helps processes move forward and extend. And that could be why it's always been associated with the reactive phenotype. Another complication with GFAP is that past HD studies are inconsistent in their use of GFAP as a marker for striatal astrocytes due to its patchy staining of the striatum. And I will expand on that in a few slides. So, GFAP has been used as an astrocyte marker in patients to show astrogliosis in HD. In this study, HD brains were stained for GFAP as a measure of astrogliosis, and then representative images from control and HD patients are shown. HD is defined by 5 grades that were established by John Paul von Tittel in the 80s, and the grading scheme reflects the progressive nature of the disease. So you can see here, the density of astrocytes increases progressively as disease severity increases. So I wanted to look at astrogliosis in our mouse model of HD. There's been some inconsistent data coming from different mouse models and at different stages of disease progression. But in some cases, like in other neurodegenerative diseases or in stroke or brain injury models, you do see clear astrogliosis. So I wondered what was going on in our mouse model. There's been a boom in a couple areas of research, and I could probably spend 40 slides telling you all about it, but I'll try to summarize it in a few sentences. First, there's been a boom in astrocyte heterogeneity research. There's been a lot of work showing astrocyte differences in astrocytes at baseline, in general, and in different regions, but also in how they respond to different insults. Second, there's been a boom in striatal function research. So there's been a lot of research coming out on different areas of the striatum being important for different functions. In mouse HD research, the striatum is usually viewed as a whole, and fairly little emphasis is put on where in the striatum you're analyzing. And this is because you get HTT aggregates all throughout the striatum. So it makes sense. But in addition, the field also tends to view astrocytes as a homogeneous population. People will select whichever astrocyte marker best fits their experiment, typically in terms of like species compatibility of the antibody. So I started with the simple question of which astrocytes in terms of marker expression increase in HD and where in the striatum do they reside. I did do this experiment with other astrocyte markers, but today I am only going to be talking about the GFAP story. Before I show you some data, I wanted to quickly explain the HD mouse model that we use. There are a couple out there, and I will talk about those later in the presentation. But in our lab, we utilize the heterozygous ZQ175 knock-in HD mouse model. This mouse model contains a chimeric mouse-human exon 1 that carries an expansion of approximately 188 CAG repeats. And we and others have fully characterized this mouse model. And it shows slow and progressive accumulation of HTT aggregates in the striatum and in other brain regions, transcriptional and electrophysiological alterations, and motor and psychiatric impairments. And in most of the experiments that I'm showing you today, I am using 12 and 18-month mice, so they are symptomatic. I just wanted to throw this slide up quickly so we're all on the same page. This is what a coronal section of a mouse brain looks like. This is the striatum, and this is the most heavily affected brain region in HD. Up here is the cortex, and that has very different roles depending on which specific part you're talking about. But this area up here where I have the word written is probably the primary motor cortex. And then lastly, between the two is this lighter piece here is the corpus callosum. Okay, so I started with GFAP, and I noticed very quickly that GFAP staining was indeed patchy. This is what people meant when they said that it had patchy staining. It does look odd for sure. However, I looked at a lot of samples, and this staining pattern was very consistent. There were little groups of astrocytes with more astrocytes up in the dorsal medial corner. So I went ahead and I started counting these cells. And in the Gomez-Pastor lab, we take 3 images per striatum, and they reflect 3 striatal locations: dorsal medial, centromedial, and centrolateral. And we typically would average these 3 locations together to get a value representing the whole striatum, as well as each individual region. And I particularly wanted to look at the whole striatum and the individual regions. Okay, let's start over here. I'll direct your attention to the whole striatum values first. As expected, there was a significant increase in GFAP-positive astrocyte density in the HD mice compared to wild type. But when you break it down into regions, it's actually only the dorsal medial and centromedial regions that are increased compared to wild type. And you can see it in these images here. It's the dorsal medial and centromedial region that are carrying the overall increase in the whole striatum because they're all up in this corner. The astrocytes are also grouped together in little clusters, which you can see in this image and here, and honestly, you can see it even zoomed out. This is unique to GFAP-positive astrocytes. I don't have time to show it, but I did look at some other astrocyte markers, and I did not see this spatial pattern. The other subtypes are pretty homogeneously distributed. So I wanted to quantify the clustering that I was seeing. In other neurodegenerative diseases, clustering of glial cells can occur around aggregates or dying neurons. So I use nearest neighbor distance, or NND, as a measure of clustering. So how it works is for each cell, so for example, the bright green cell in this image, you measure the distance from its center to the center of every other cell in the image, and you find the nearest neighbor, which in this case would be this cell, and therefore the nearest neighbor distance is 331 microns. In this case over here, you do the same thing, measure the distance to every other cell in the image, and you find your nearest neighbor, which is 218 in this case. And then you do that for every single cell in your image, and you get an average NND, where the smaller the average NND, the closer together cells are. However, NND alone does not technically account for increased density, since having more cells in the same area would also decrease NND. So I calculated spacing index, which is NND squared times density, and therefore does take into account density changes. And when I ran this analysis, I saw that NND and spacing index were both significantly decreased in HD mice compared to wild type, meaning that the GFAP-positive astrocytes are significantly more clustered in HD mice. You can see it here. The astrocytes are also showing hypertrophy here, but the measurement is from the center of each astrocyte, so that shouldn't change anything. The other subtypes I tested did not exhibit clustering. They had a more random distribution. So the next logical question is, what are these cells clustering around? I thought it could be areas of high aggregate load, areas with a lot of inflammation, or possibly even areas with dying neurons. So first, I looked at GFAP and EM48. EM48 marks HTT aggregates. And I looked at 5 different time points, 3, 6, 12, 18, and 22 months. GFAP positive astrocytes increase over time. There really aren't very many at 3 or 6 months, but they do start to accumulate, and there's a ton by the time you get to 22 months. EM48 aggregates also increase over time, with aggregates appearing around 6 months and then increasing in number and size as disease progresses. This is well known in the field, and it's been established in our mouse line. If you look at EM48 staining, you can see areas of the image where there are less aggregates, like here. I'm kind of outlining a region where I see a lot less dots compared to out here, for example. And I noticed that occurring throughout the striatum. When I looked at GFAP and EM48 together, I saw something really interesting. It appears that GFAP positive astrocyte clusters match up perfectly with areas of low aggregate load. So on this slide, I just outlined the clusters by hand, but I actually did extensive image analysis where I created perimeters that were 10 microns away from the edge of a GFAP positive pixel. And then when astrocytes are close enough together, the perimeters kind of merge and it ends up outlining an entire cluster. So I was able to count EM48 puncta inside versus outside of the clusters, and I saw significantly less aggregates in the inside GFAP clusters. So I got really excited because maybe these astrocytes are clearing aggregates. That has been seen in other neurodegenerative diseases, but something wasn't adding up. If you look in different striatal regions, EM48 puncta counts do not significantly differ. I added a 4th striatal region here, which will pop up in future data as well, but it is just DL, which stands for dorsolateral. Anyway, these holes in EM48 staining are present in all regions too. So it doesn't make sense because GFAP positive astrocytes are only in that dorsal medial corner. There must be something else to explain that connection. So I next turn to some anatomical explanations. 2 things that are larger in the dorsal medial striatum compared to the centrolateral are 1, striosomes, and 2, white matter fascicles. Striosomes are compartments with different neurochemicals than the surrounding matrix, but they're pretty big. They're definitely bigger than the holes that I was seeing in EM48 staining. So I looked at white matter fascicles, which appear to be a better candidate based on size. To simplify for those not in neuroscience, here's your neuron. You have a cell body here, and then an axon that is surrounded by myelin sheaths, which help action potentials propagate down the axon. The myelin is made up of mature oligodendrocytes that wrap around the axons. And when we say gray matter, we're referring to areas of the brain that mostly contain cell bodies, whereas white matter primarily consists of myelinated axons. Cortical neurons have cell bodies sitting up in this gray matter, and some of them send projections to other brain areas. So many axons will join up together and they travel as groups, and that's white matter. Sometimes they travel in really large groups like the corpus callosum, which is a white matter structure that crosses hemispheres. Or sometimes they just travel in bundles of a few axons, and those would be called fascicles. In humans, the striatum is separated into the caudate and the putamen, and they are separated by the internal capsule, which is a bundle of white matter traveling to and from various brain regions. But in rodents, there is no separation of caudate and putamen. Instead, they're one structure with white matter fascicles running perpendicularly through it. And I've shown here just a simple light microscopy image where you can see the cross-section of white matter fascicles among the gray matter of the striatum. So in humans, you have distinct gray matter, white matter, gray matter, whereas in mice, it's kind of a mix of gray matter and white matter together. So I stained for GFAP and MBP. MBP is myelin basic protein and therefore stains white matter. And I am showing just the wild type brain first so we can get oriented. This is a coronal section. This is the only orientation I've shown you so far. But let's talk about MBP specifically. I am showing that here in pink, and you can see those fascicles that I was talking about. There's tons of cross sections of fascicles throughout the entire striatum. Now I'll add those HD mice. So I added another coronal section here, but this is a sagittal section. And in the sagittal view, you can see those fascicles long ways. GFAP is shown in green, and hopefully you can appreciate that the GFAP positive astrocytes appear to be associated with white matter fascicles passing through the striatum. Specifically over here, I think is a really good image of this. And here's some zoomed images that are even better. They basically show the same thing. The GFAP positive astrocytes are either surrounding or within the white matter fascicles. And this occurs even in wild type, just with less GFAP positive astrocytes. And this makes a lot of sense. GFAP is actually highly expressed in white matter astrocytes. This has been known for a while, but typically HD researchers think of the striatum as a gray matter structure, when in reality, it has a ton of white matter. But what's really interesting to us is that there is selective involvement of some fascicles and not others. So like in this image up here, you can see that these fascicles have no GFAP positive astrocytes nearby, and this occurs in wild types as well. But the other really important thing is that all of these fascicles have a low aggregate load in HD mice. You can see that there's very few pink dots in the area of the fascicles that I have outlined, regardless of whether they're GFAP positive or GFAP negative. To dig a little bit deeper into the fascicles themselves, we counted up fascicles and measured their size. So up here, I am showing that the number of fascicles is not significantly different in any region of the striatum. Down here, we looked at fascicle size throughout the whole striatum, and we broke them up into size categories, small, medium, and large. And we did not reach significance here, but there is a possible trend towards smaller fascicles in the HD mice, towards more smaller fascicles. I also separated the fascicles out into GFAP positive or GFAP negative fascicles. So first I counted them and I saw significantly more GFAP positive fascicles in HD than in wild type in the dorsal medial and dorsolateral regions only. And then I measured fascicle area, and I found that GFAP positive fascicles were smaller in HD mice than in wild types. But overall, GFAP positive fascicles are way bigger than GFAP negative fascicles in both wild types and HD mice. Now, I don't want to neglect the white matter changes in HD that have been documented for decades. In human HD brains, it's been shown that there is white matter pathology, such as reduced white matter volume, as shown in this graph from an imaging experiment with HD patients. There's also thinning of the myelin sheaths and myelin breakdown in HD. It's also been shown that there is lower aggregate load in the white matter compared to the gray matter. This is an image that is showing aggregate staining in HD mice. And you can see how the corpus callosum and the white matter fascicles of the striatum, which are the white holes here, have much less staining than the gray matter of the striatum or the cortex. So it's not necessarily surprising that we see a low aggregate load in white matter tracts, regardless of GFAP positive astrocyte association. While the white matter pathology in itself could certainly be an explanation for why GFAP positive astrocytes are near white matter fascicles, I come back to the fact that these GFAP positive astrocytes are specifically near certain fascicles only those ones in the dorsal medial corner, and even there, only some of them. So why just some of them, and which ones are they? So with those questions in mind, I teamed up with Sarah Heilbronner's lab. She used to be faculty here, but has since moved to another institution. We were a bit out of our depth, and we really needed some guidance on where to go next. And really what we wanted to know was which area of the brain sends fascicles through the dorsal medial striatum, particularly in these areas where we see GFAP accumulated. So her lab was able to help me narrow down candidates using the Allen Brain Connectivity Atlas. And this is a series of experiments where fluorescent and pterograde tracers were injected into different brain regions. So basically, they inject this virus into a certain brain area and it causes the whole cell to fluoresce, including its axon, as it travels. And so you can basically follow the path of neurons from their point of origin and see where they go and where they terminate. And that's the idea here. There was an initiative to do this in basically every brain area and image everywhere. And this helps us understand how different areas of the brain are connected. And I used this collection of data to help me work out which fascicles are passing through the striatum. So we looked at experiments with injections into the primary motor, or M1, anterior cingulate, infralimbic, and prelimbic regions. And all of these regions send fascicles through or to the striatum, but not in the dorsal medial corner. They're either too lateral, too medial, or too ventral. And this one's also too ventral. But then we looked at M2. M2 is the secondary motor area in rodents. And I have my image on the left here with the region of high GFAP positive astrocytes accumulation boxed. And then on the right, I have an image from an Allen Brain Connectivity Atlas experiment that shows pterograde tracing from M2 passes through the very same region. Here I have more images from 3 different tracing studies in that connectivity atlas. You can see on the left side, there's injection location, and then on the right is where they pass in the striatum, which are all in the dorsal medial region. And in this first one here, you can actually see the fascicles coming off of the corpus callosum, which is pretty cool. So based on this, we think that M2 fascicles are a pretty good candidate. And so I did the tracing myself. Here I am showing my injection location in M2. I used a GFP virus. And when I do that, basically all the M2 neurons are going to be labeled with GFP, and you can follow the axons of these M2 neurons. And when you do that, you can see them in bundles in the dorsal medial striatum. Here and here, you can actually see them coming off the corpus callosum and forming those bundles as they pass through the striatum. Here I stained with MBP in pink, and you can see that those M2 fibers are indeed part of white matter fascicles. And they're actually part of big white matter fascicles, which is great news since we previously saw that the GFAP positive fascicles tend to be large. And then separately, since at the time I did not have a compatible antibody, I stained for GFAP, and you can see that these M2 fibers do have GFAP positive astrocytes associated with them. An important note here is that this particular injection was done in one singular 12-month mouse, so I did not quantify anything, but I have since done more injections and I will be able to quantify those, so stay tuned. But anyway, over here are some more zoomed images that show clear association of GFAP positive astrocytes with M2 fibers. Here is an animation, hopefully it plays. You can see in 3D that the pink astrocyte processes are really interweaving through this fascicle, and they're in close proximity to these fibers. I was recently presenting this work at a conference, and someone who works with axon injury said that these axons didn't look very healthy. So that is something that we will be looking into as well. Are M2 axons unhealthy in HD, or was it just my virus? So I would like to summarize what I've told you so far. HD mice have more GFAP positive astrocytes than wild types, and they're all located in the dorsal medial corner of the striatum. They appear to be associated with low aggregate load, but that was explained by white matter. GFAP positive astrocytes are associated with white matter, and white matter has a low aggregate load. And most importantly, GFAP positive astrocytes are associated with specific white matter fascicles in the dorsal medial striatum, and they're most likely coming from M2. So let's talk about M2 for a second. M2 is an interesting brain area. I'm still learning a lot about it, but there have been numerous studies based in anatomy, lesion studies, electrophysiology, and connectivity that show M2 is a possible homologue for the primate premotor and supplementary motor areas. M2 receives inputs from tons of brain areas, including sensory input, and it projects to many areas as well, including some associated with motor control. And many of M2's connections are reciprocal, actually. It's likely that M2 plays a role in linking sensory cues to motor actions, and therefore in the flexible control of voluntary actions. And since I figure this is going to be a question, I will just get ahead of it. Yes, there have been a few HD studies that looked into these regions. Overall, cortical pathology is hugely debated in the HD field. However, there are some broad studies that look at a lot of different areas and happen to have some data for these regions. We know from imaging studies that there is cortical thinning in these two areas, and we know from a postmortem study that there is a decrease in pyramidal neuron number in the premotor area. And interestingly, it seems like both of these things are affected by symptom type. So patients can have more motor symptoms, or they can have more cognitive symptoms, for example. So it might not be seen in all patients, and maybe that's why there's been a lot of debates about this. And I don't believe any study on aggregate load was specific to these two areas. Since I'm trying to figure out exactly where fibers that pass through the striatum are coming from, I need to be a little bit more specific. Since M2 is doing a lot, we need to look at individual layers. So the cortex is organized in layers, which are defined by cells with different sizes, shapes, inputs, and outputs. And in M2 specifically, layers 2 and 3 project to other areas of the cortex. So it's not going to be them. But layers 5 and 6 do project through the dorsal medial striatum. Both of these layers have projections that go elsewhere, but I am strictly talking about the fibers that pass through the dorsal medial striatum. So layer 5 fibers go to the spinal cord as part of the pyramidal tract, and layer 6 fibers go to the thalamus. And based on some images that I did not show today, in which I did serial sectioning of the whole brain, and I followed where there was an abundance of GFAP positive astrocytes in HD brains, and it appears that the pyramidal tract is involved. So my guess is that the important layer is layer 5, and that is more relevant to me, but we'll still test both layers 5 and 6 in the future. Regardless of where these fascicles originate, one important question is, what are these GFAP positive astrocytes doing? Astrocytes interact directly with both axons and with myelin sheaths. I showed you earlier that there is evidence for myelin breakdown in HD. It's known that stressed myelin can release factors that cause inflammation, and that can cause astrocytes to become reactive and express GFAP. On the other hand, it's been shown that in a lot of demyelination models, that astrocytes are actually very important for remyelination. So they could be there for that reason. The other alternative relates to axons. First, many axonal injury studies have shown that astrocytes show up after axonal injury to clean up debris. But second, astrocytes can actually surround a node, and a node is the region in between individual myelin sheaths on an axon. And the astrocytes can influence conduction speed of axons by releasing ATP. That's pretty new research, so there's not a ton about it, and there's definitely not much on how that interaction is affected in neurodegeneration or demyelination cases. And I'm sure I'm missing some other possible explanations of interactions, but these are just some that I've been thinking about. So both their interaction with myelin and with axons are things that I think are very important to look into. So where do I go from here? I definitely want to dive into M2 a bit more. First of all, I have a bunch more mice that I did tracing studies on, so we can actually quantify some of that data. I also plan to explore the layer specificity a bit more. There are promoters that are layer specific, so I will be looking into that. We're also looking at M2 pathology. We want to know what's going on there. So we're going to look at glia, aggregates, MBP, cortical thickness, and layer size. Next, we want to look a bit more into the myelin. So I did not show all of this today, but I did not see difference in OLIG2 positive cells. Those are oligodendrocytes. So I didn't see differences in oligodendrocyte cell numbers or in overall MBP intensity. I did show you that I didn't see any differences in fascicle size. But despite all that, any real myelin researcher would tell you that you need to do EM. And so that's what we're going to do. You can do something called a G ratio to assess myelin thickness, and you can look for irregular myelin patterns. And then lastly, I think we should really check on those M2 neurons and their electrophysiological properties. There is a huge possibility that these neurons are dysfunctional, and that's why a bunch of astrocytes are coming over to help. So we'll definitely check that out. And there's just a lot of cool stuff to follow up on. Okay, so now that I told you about my project, I wanted to have a little bit of a discussion about mouse models and HD research. And I will give a spoiler that my research has some questionable translatability simply due to species differences. Mouse and human astrocytes are different, rodent and primate brain anatomy is different, and HD mouse models aren't perfect. But we'll go through each of those. I don't want it to come off like I'm throwing the validity of my research into the trash. I just think that these are really interesting discussion opportunities and something that I am going to continue to have to think about in a future neuropathology and neurodegeneration research career. Okay, human astrocytes are larger and more structurally complex than their rodent counterparts. There's also some evidence that human astrocytes are even more diverse than mouse astrocytes, which are already pretty diverse. One lab directly tested astrocyte responses to a variety of insults, such as oxidative stress, hypoxia, inflammation, and infection, and they found significant differences in human versus mouse astrocyte pathology in their responses. And this is really important since neurodegenerative diseases like HD have oxidative stress and inflammation components. So if we can't model that correctly in mouse models, that could be a huge issue. I also would like to mention here that I am mostly talking about in vivo things today, but there are plenty of in vitro challenges when it comes to astrocytes. And using cultured human astrocytes is actually really hard to get right. Okay. Next, I wanted to revisit this concept because it poses a challenge for translating my findings to humans. I'm telling you that in my mice, I see GFAP positive astrocytes accumulating within and around specific white matter fascicles as they pass through the striatum. However, that is just not something that we're going to be able to see in humans. I won't see individual fascicles because those fascicles are part of the internal capsule in humans. And even though there is some organization, and even though there is some organization in terms of where the fibers are in the capsule, there's no overlap and there's no distinct borders, especially when talking about fibers originating from smaller and more specific cortical regions. Now I'll bring this slide back up. This is showing GFAP positive astrocytes in the striatum of human brains. And clearly this was not what I was seeing in my mouse model. In patients, GFAP positive astrocytes are present and progressively increasing in the gray matter as disease progresses. It's even a criteria in the grading scheme. But I don't see gray matter GFAP positive astrocytes in my mice. All of the GFAP positive astrocytes I see are associated with white matter. White matter astrocytes are commonly GFAP positive. And that is true in humans and in mice. Here I have a picture of white matter from a controlled patient, a brain that is stained for GFAP. And over here, I am showing you a wild type mouse. I showed this picture earlier, where there's tons of GFAP in the corpus callosum, which is a white matter structure. And so there is a lot of GFAP in the white matter normally. This means the internal capsule in humans is going to be full of GFAP positive astrocytes unrelated to disease state. So if you add that to the problems we discussed in the last two slides, you can't differentiate if specific bundles of axons are GFAP positive or not. So to summarize this part, it's going to be nearly impossible to see if GFAP positive astrocytes are doing similar things in humans and mice. But if the GFAP positive astrocytes are alerting us to some kind of pathology or disturbance in M2, that will be something that we can look into in both mice and humans for sure. So the last thing that I wanted to talk about while we're on the topic of translatability was the mouse models that we use in HD research. I'm just going to focus on the most popular ones here, but there are plenty of others. So the CAG expansion in Huntington disease in the Huntington gene is in exon 1. So some of these models have just exon 1 and some have full-length HTT. I'll start with the R62s first. These are a transgenic model that express exon 1 with approximately 120 CAG repeats. The yak in the back models are full-length HTT inserted into either a yeast or a bacterial artificial chromosome. And the benefit of that is that you get full-length HTT, but you also get protection from other mouse DNA and any positive or negative effects that it might have on expression levels. The CAG140 and ZQ175 mice are knock-ins of exon 1 with 140 and 175-ish repeats. And that is knocked into the endogenous mouse Huntington gene. You may notice that all of these repeat numbers are extremely high. The first models attempted to use smaller repeat numbers that better reflected patient repeat size, but those mice showed extremely mild behavioral phenotypes, if any at all. So we use these models with higher repeat numbers in order to see phenotypes. The next important difference is in CAG tract purity. The CAG tract is prone to expansion and instability, and for that reason, mice with pure CAG repeat tracts need to be monitored occasionally for repeat length. For example, the CAG140 mice were originally developed, and then the colony underwent a spontaneous expansion to 175 repeats, giving rise to that ZQ175 mouse model. But by the time that mouse model stabilized, it actually had closer to 188 repeats, and that's what I told you earlier. But we actually just checked our colony last year, since you do have to check occasionally, and I think we were averaging around 165 repeats. So again, just emphasizes the instability. But on the other hand, the yak and the back models, they have mixed CAG and CAA repeat sequences, and that helps with the stability of the repeat size. CAA also codes for glutamine, so it doesn't change the protein, but it does help keep the repeat number stable. Another important difference is in disease progression. Most of these mouse models are slow progressing with mild phenotypes. They'll live the full 2-year mouse lifespan, and they'll gradually show more and more symptoms. But the R62s, on the other hand, are a rapidly progressing model. Those mice show motor symptoms as early as 5 to 6 weeks, and they usually die by 16 weeks. And no one really knows exactly why that model is so rapid. Lastly, the back HD mouse model has a floxed exon 1, which again is where the repeat expansion is. And so you can use cell type specific Cre viruses to remove mutant Huntington in certain cell types only. So that's a really cool thing about this model, and it actually helped to do many of those cell type specific experiments that I showed at the beginning. There are other animal models, but they're rarely used in the HD field. There's a fruit fly model, a back HD rat model, and then recently there's been attempts to move into large animal models, but there's been a variety of problems. Those animals are often not surviving, and then when they do, they're not recapitulating human disease very well. So those are just some of the models. I'm definitely leaving out some of the lesser used ones, but the key differences are in whether they're transgenic or knock-ins, how fast the disease progresses, CAG tract purity, and then whether or not it's floxed. As for similarities, these mice never really get to the same disease endpoint as humans. Even the most severe mouse model, the R62s, we don't see the same motor impairments that we see in humans, and we definitely don't see the MSN death and degeneration in our mouse models. And that's with extremely high repeat numbers, way higher than anything we see in humans. So these are big things to be missing, but nobody has been able to create a model that fully recapitulates those aspects of human disease. Now, this is not to say that these models aren't useful. Obviously, they are. We've learned a lot about HD from mouse models. Almost everything that we've learned about early manifestations of this disease have come from these models, and they're used to test possible therapeutics and interventions. All of the research on electrophysiology of MSNs and calcium signaling has come from these mouse models too. So super useful things that we could not have learned from patients. However, it does pose limitations in the questions you can ask with these models. For example, you wouldn't want to ask questions about repeat length stability and mechanisms of expansion in the YAC128 or the BAC-HD mice, and you wouldn't want to answer questions about cell death mechanisms in models that don't exhibit much cell death. Modeling HD is a real issue. Now, I'm not even going to get into the in vitro options, but suffice it to say that it's been a challenge for the field. In a disease where so many cell types are affected and they're all influencing each other, plus the added disease progression over time, there's a lot of factors at play that may or may not be represented in whatever model you choose. So are there translational challenges in studying HD? Absolutely. And does HD astrocyte research in particular come with a lot of challenges? Definitely. But for me and my research, it could go two ways, or at least that's how I view it. First is that what we're seeing in astrocytes in HD mouse models doesn't translate at all. And if that's the case, why it doesn't translate is a huge question. Can we continue studying rodent astrocytes and expect them to behave or respond the same way in humans? Maybe it's different for different brain areas or disease states. Who knows? But the second option is that what we're seeing in astrocytes in our HD mouse models does translate. And it's really, really hard to see in humans, but really easy to see in mice. And that's when species differences works to our benefit. So these are the messages I wanted to convey today. There are a lot of challenges, and there's definitely room for improvement. But as a field, HD research has come a long way while using mouse models. And they found out a ton about early gene dysregulation and cellular dysfunction that we would have never been able to see in patients. So these models are extremely useful. And while they might not be able to fully recapitulate human disease, they've been absolutely essential so far, and they're playing a huge role in helping us uncover the underlying mechanisms of HD. So that is the note that I would like to end this presentation on. So thank you for listening, and thank you to all of these people for helping with the project and with my progress so far. And now I can take any questions.
So are there any questions? I have some if no one else does. At what age were most of these studies done? How old were these mice?
Yeah, so the beginning ones where I was doing cell counting are 12 months, and then I moved to 18 months for pretty much everything else you had seen.
Okay. So what does the striatum look like at 18 months in terms of neuronal populations? Is there a loss of cells, or is there at least a volumetric shrinkage of the structure?
Yeah, that's a good question.
I'm not familiar with this model particularly, but
yeah, we usually I think we didn't do 18 months before I started, so it's 12 and 22 that we looked at. We do not see any differences in cell number. Actually, all we see is differences in transcription. So it seems like these cells are not dying, but they are transcriptionally dysregulated. We never see full cell death, and then we don't see any volumetric changes either. And that's, again, characteristic of pretty much all of these mouse models. That's kind of the endpoint that we see.
And Dr. Crossan has a question.
Dr. Crossan. John?
Taylor, that was a fascinating talk. Thank you so much. I was particularly interested in some of your comments about the astrocytes. And you obviously mentioned that astrocytes are very diverse. And I was wondering if the GFAP is a marker for a single subset of astrocytes with specific characteristics, or is there a lot of overlap? And are you able to separate these different diverse astrocytes using certain markers?
That's a good question. The answer is kind of complicated. it seems like GFAP is highly expressed in white matter astrocytes. So that's number one. But it's also turned on when astrocytes are reactive. And so that will happen in gray matter astrocytes that become reactive. But it turns out that's also kind of on a spectrum. And so you can find different genes turned on at different points in that spectrum in terms of reactivity. So realistically speaking, they're probably not marking only one subtype of astrocyte. They're probably marking more, but that's the benefit of using some kind of spatial characterization to qualify where they're located, I guess, because that gives me a better idea that those are probably white matter and not necessarily reactive astrocytes.
Thank you very much. Very interesting.
So I have another question about your the spacing index. So
Yeah.
the cells that are positive with the Huntington marker, what cells are those? Are can you tell? Are they neurons or are they glia and neurons?
Yeah, it's mostly neurons, but there are aggregates in glia. Nobody's tested glial subtypes for whether or not they have like specific subtypes have aggregates. I think probably they're just looking at astrocytes in general in those experiments, which means, as we know, it could be any subtype.
Yeah, so I guess my question is there some like volumetric loss of the neurons themselves so that cells are smaller so that things are crowding?
Yeah, we haven't seen that.
Okay. Did you look like with GAD or something like that? These are, I mean, the spiny neurons are GABAergic. I mean, you're probably, that's not really what you're interested in right now, I guess, but I just wonder, when people have looked at that, what do they see in this model?
Yeah, that's a good question. I don't know the specifics because honestly, once I figured out they were white matter, I kind of stopped digging over there. So there certainly could be what you're saying, like the individual cells are a little bit smaller, but I don't think that anybody has gone that far in depth with the mouse models, knowing that, again, that they're just not recapitulating things as perfectly.
Yeah, Chris has a question.
Yes, does the aggregated protein, is that taken up by the astrocytes, or the astrocytes responding to damage to the neurons that is caused by the aggregated protein?
Yeah, that's a great question, and we don't know the answer yet. A paper came out maybe last year that did show GFAP positive astrocytes were important in aggregate clearing, but it was all in vitro. So we have not been able to show that really in vivo. There's so few aggregates in actual astrocytes if you do kind of imaging in 3D and putting it together, that we really haven't been able to like make that claim official, but people are interested in that.
And then I just have another question about GFAP. As I recall, one of your images showed that there was an increase by 2 seconds in staining. Was that something you showed earlier? I guess my question is, if that's true, is the protein being released by intracellular stores, and is that why it popped up so quickly, or what?
Yeah, I think that was just a color confusion thing. So that was unrelated to GFAP. That was a calcium indicator over time.
Oh, yeah. I'm sorry. That's right. Yep. I forgot about that. Great talk, by the way. Thank you.
Thank you.
Jim, you have a question?
I do. Taylor, that was a terrific talk.
Thank you.
I know very little about the brain, but my field is in extracellular matrix. And the extracellular matrix in the brain has been fairly well characterized. It's rich in hyaluronan and a lot of other proteoglycans. And so when you're looking at spatial interaction or spatial relationships, especially in terms of inflammation, because a lot of the matrix will start to become degraded and change in composition. Have people looked at that in Huntington's to see whether or not there are changes that start to change positions of cells because the matrix that they're embedded within is changing? And perhaps there's no answer to this, but something.
So that's a great question. I do not know the answer to it, but it is definitely something I will look into.
Again, you did a great job. Thank you so much.
So are there any other questions? We're just about out of time. Thank you very much, Taylor. That was a great talk, and looking forward to having another neuropathologist join.